Good morning, students. Welcome back to Microbial Nutrition Part 3. Myself, Dr. Ragasudha, Assistant Professor in Government City College, Hyderabad, is here to discuss on uptake of nutrients as a part of microbial nutrients. In order to support its activities, a cell must bring in nutrients from the external environment across the cell membrane. In bacteria and archaea, several different transport mechanisms exist. The nutrients must pass through a selectively permeable plasma membrane, will not permit the free passage of most of these substances. Nutrient molecules frequently cannot cross this selectively permeable plasma membrane through passive diffusion and must be transported by one of the three major mechanisms involved in the usage of membrane carrier proteins. The four major types of modes of nutrition observed in microorganisms are passive transport or simple diffusion, facilitated diffusion, active transport, and group translocation. Passive diffusion. A few substances such as glycerol, water, oxygen can cross the plasma membrane by passive diffusion or simple diffusion. Passive diffusion is a process in which molecules move from a region of higher concentration to the one of lower concentration as a result of random thermal agitation between the molecules. This is an example for simple diffusion where we can see the diffusion of molecules across the cell membrane from the external cellular spaces to the internal cellular spaces, and once an equilibrium is attained, the diffusion ceases. So passive diffusion is diffusion of very small molecules like water and non-polar gases like CO2 across the plasma membrane. It does not carry any protein or energy. The movement is along the concentration gradient and requires high external concentration. If concentration gradient disappears, then net inward movement stops. This mechanism is reversible and it is non-specific as there are no carrier protein molecules. Proteins, and it shows saturation. The process is slow and thus may not extensively employed in the organisms. Moving on to the second type, facilitated diffusion. The rate of diffusion across selectively permeable membrane is greatly increased by use of carrier proteins, sometimes called permeases. These are embedded in the plasma membrane. Since the diffusion process is aided by a carrier, it is called facilitated diffusion, indicating that a facility is provided for the transport of molecules from outside environment into the cell. The rate of facilitated diffusion increases with the concentration gradient. It is much more rapid when compared to passive diffusion. Here the graph between rate of solute energy and external concentration of the solute is plotted, and the difference between simple diffusion and carrier mediated transport or facilitated diffusion is shown. We can see a very good amount of difference between both the mechanisms. So in facilitated diffusion, the permeases are embedded in the membrane in this fashion, and these permeases bind to the outside solute particles or nutrients on the outer end, and they are transported along the permeases into the cell. The rate of diffusion increases with the concentration gradient in facilitated diffusion, and it is rapid at lower concentration. Major intrinsic proteins facilitate diffusion of small polar molecules. Facilitated diffusion allows lipid insoluble molecules, and a good example of facilitated diffusion in bacteria is transport of glycerol. Characteristics of facilitated diffusion. It involves the use of permeases embedded in the plasma membrane. It moves along the concentration of the gradient. No metabolic energy is required. If the concentration gradient disappears, the net inward flow of molecules ceases. The mechanism is reversible if internal concentration is high. These permeases show high specificity, and the mechanism shows saturation also. So the facilitated diffusion involves movement of molecules from higher concentration to lower concentration. Each carrier is selective and will transport only closely related solutes. It seems that this mechanism is not important in prokaryotes while it is prominent in eukaryotic cell. This is the model of facilitated diffusion. The membrane carrier can change conformation after binding to an external molecule and subsequently release the molecule on the cell interior. It then results in the outward orientation position and ready to bind to another solute molecule. Because there is no energy input, molecule will continue to enter only as long as there is a concentration gradient on the outside. Moving on to the third type of uptake of nutrient mechanism, active transport. Active transport is the transport of solute molecules to higher concentrations or against a concentration gradient with the use of metabolic energy input. So unlike passive diffusion and facilitated diffusion, here the movement of molecules are from lower concentration to higher concentration with the use of metabolic energy. Characteristics of active transport. It involves the use of permeases. It is against concentration gradient and the metabolic energy is required for the process. It uses metabolic energies like ATP, high energy phosphate compounds or proton motive force. It shows saturation and the permeases show specificity for the molecules being entered, and this mechanism is highly irreversible. A good example for active transport system is ATP binding cassette transporters, or in short it is known as ATP ABC transporters. They are observed in all the three domains of life. They contain two hydrophobic transmembrane domains, each associated with an ATP binding domain on the cytoplasmic side. The transmembrane domains form a pore in the membrane that allow substances to be transported across the cell membrane. The ATP binding domains bind and hydrolyze ATP to ADP that provides energy for the transport against a concentration gradient. The ABC transporters employ special substrate binding proteins present in the periplasmic space in gram-negative bacteria, while in gram-positive bacteria they are found attached to the membrane lipids on the external face. Several superbugs are able to expel drugs out of the cell by ABC transporters. Another example for active transport is proton gradient. Beside using energy from ATP, ATP is a primary mechanism of active transport, whereas proton gradient is a secondary mechanism of active transport. So here they can harness energy of the proton gradient that is established during electron transport chain. Proton gradient shows three mechanisms of transport of substances. They are symport, antiport and uniport. Symport stands for linked transport of two substances in the same direction, while antiport stands for linked transport of two substances in the opposite direction, whereas uniport is for transport of a single substance into the cell. So here we can see all red colored molecules are transported molecules, whereas yellow are co-transported molecules. Yellow and blue are co-transported molecules. So in uniport we can see one molecule entering the cell, whereas in symport we can see red and yellow two molecules being transported into the cell simultaneously. In antiport we can see that the red molecule is entering the cell, at the same time blue is exiting the cell. In proton gradient, one example in E. coli is uptake of lactose by lactose permeases. This permease is a single protein that takes in a molecule of lactose along with a proton. This kind of transport is known as symport, wherein lactose and a proton are moving inside the cell simultaneously. This picture shows the proton gradient, and along with the proton gradient, when the hydrogen ions are being pumped out of the cell with the help of ATP, while these hydrogen ions re-enter the cell by co-transporting sucrose. This is known as symport. When lactose and proton bind to their respective sites on the permease on the outward facing conformation, the transporters change conformation, now face towards inside. This reduces the binding affinity of the molecule to their sites and consequently are released into the cytoplasm. E. coli uses this energy in proton gradient also to take up amino acids and organic acids like succinate and malate. The second type of proton gradient is antiport. Antiport can also power active transport indirectly through a sodium ion gradient. In E. coli, a sodium ion moves out of the cell as a proton moves in. This sodium ion gradient is then used to take up amino acids and sugars. It goes to a carrier protein. The carrier protein then binds to the substrate of interest and orients towards to the interior of the cell. Both of them then dissociate subsequently. E. coli uses this form of transport to transfer mallebiose and glutamate. It is also present in eukaryotes. The fourth mechanism of uptake of nutrients in microorganisms is group translocation. It is a process in which a molecule is transported into the cell while being chemically altered. The best known group translocation system is phosphoenol pyruvate sugar phosphotransferase system, abbreviated as PTS, which transports a variety of sugars into prokaryotic cells while simultaneously phosphorylating them using phosphoenol pyruvate as the phosphate donor. This is the equation showing how phosphoenol pyruvate phosphorylates the incoming sugar molecule. PEP is phosphorylates the outside sugar and converts into pyruvate, and the sugar is converted into sugar phosphate. So this particular system involves the following components. Number 1 is phosphoenol pyruvate. Number 2 is enzyme 1, which is a low molecular weight heat stable protein present in the cytoplasm. Enzyme 1 is common in all PTS systems. The third component is enzyme 2. This enzyme 2 has 3 subcomponents or subunits: enzyme 2A, enzyme 2B, enzyme 2C. Enzyme 2A is present in the cytoplasm and it is soluble in nature. E2B is hydrophilic but frequently found attached to E2C. E2C is a hydrophobic protein that is embedded in the membrane, which allows the transport of sugar molecule from outside to inside of the cell. E2 enzymes are only specific for sugars and it varies with different PTS systems. Here in the picture we can see the phosphoenol pyruvate sugar phosphorylase system of E. coli. The phosphorylate group on PEP is transferred to the imported sugar via several proteins, that is enzyme 1 and enzyme 2. The phosphoryl group is first transferred to enzyme 1, a heat stable protein. Then it is transferred to enzyme 2A, where it is phosphorylating a histidine residue. Then it is transferred to enzyme 2B, where the phosphoryl group is transferred to a cysteine residue. Finally, this enzyme 2B phosphorylates glucose or mannitol as it crosses the plasma membrane through the transmembrane enzyme 2C found embedded in the membrane, forming glucose 6-phosphate or mannitol 6-phosphate. So what is the benefit of phosphorylation of incoming sugar molecules? The benefit of transfer of glucose to glucose 6-phosphate or mannitol to mannitol 6-phosphate is to prevent leak out of the cell. The glucose phosphotransferase system is ultimately regulated by the E2 enzyme. Lower concentration of glucose phosphorylate E2 phosphorylates E2A and stimulates the process, while high concentration of glucose dephosphorylates the E2A enzyme. Now let us see a simple comparison of all the four transport systems. Carrier proteins are absent in passive diffusion, present in facilitated, active and group translocation. The transport speed is slow in passive diffusion while it is rapid in facilitated, active and group translocation. The movement of molecules is against concentration gradient in active and group translocation, while it is along concentration gradient in passive and facilitated diffusion. The transport of molecules is non-specific in passive diffusion and specific in facilitated, active and group translocation. Metabolic energy is required for facilitated, active and group translocation, whereas it is completely absent in passive diffusion. The solute molecules are not changed in passive diffusion while they are changed during the transport in facilitated, active and group translocation. Moving on to another important uptake of nutrients, iron. Iron can act as a very essential micronutrient to microorganisms for use in cytochromes and many enzymes. Iron uptake becomes essentially difficult due to its insoluble properties, which leaves little free iron for utilization. Hence, several bacteria and fungi secrete siderophores. Siderophores are low molecular weight organic molecules that can complex with ferric iron and supply it to the cell. 3 molecules of siderophores complex with one ferric iron atom via 6 coordinate bonds. Siderophores are of two types, hydroxymates and phenolate catecholate. Hydroxymates example ferrichrome found in fungi. Phenocatecholate example is enterobactin produced by E. coli, while both the systems are produced by cyanobacteria. In the picture we can see both the systems present in cyanobacteria. Hydroxymate siderophores, 3 siderophores bind to a single ferric ion and transports it into the cell, transferring into ferrous ion. Similarly, catecholate, one catecholate binds to the ferric ion outside the cell and transports and converts it into ferrous iron. Microorganisms secrete siderophores when iron is scarce in the medium. Once the iron siderophore complex reaches the cell surface, it binds to a siderophore receptor protein. Then either the iron is released into the cell or the whole complex is transported by a ABC transporter. After the iron enters the cell, it is reduced to ferrous state. So these are the siderophores with receptors which bind to the externally present ferric ions, and these are transported into the cell along with the into the siderophore and gets transformed ferrous iron. Summary. In the first video, we have discussed about nutritional requirements of microorganisms, where we discussed about various nutrients required by the organisms. These nutrients are substances in the environment used by organisms for catabolism and anabolism. The nutrients are of 3 types: macronutrients, micronutrients and growth factors. Along with these nutrients, microorganisms also require environmental factors which influence the growth of the microorganisms. In the second video, we have discussed about nutritional types of microorganisms, where we have discussed about 3 different groups of microorganisms: autotrophs, heterotrophs and mixotrophs. Autotrophs are the organisms which have produced organic compounds from simple inorganic compounds using light or chemical energy. Heterotrophs require organic substances to get chemical energy for its growth and development. Mixotrophs can use both the above modes of nutrition. And these mixotrophs are of 2 types: methylotrophs and methanotrophs. Methylotrophs are the organisms that can grow using carbon compounds that lack CC bond, while methanotrophs metabolize methane as their sole source of carbon and energy. These organisms also require single carbon compounds for survival. In the current video, we have discussed about uptake of nutrients, wherein we have discussed about passive diffusion, which is due to thermal agitation of molecules, facilitated diffusion, wherein the movement of molecules is facilitated with the presence of permeases present in the plasma membrane, embedded in the plasma membrane. Active diffusion is associated with permeases and energy. Group translocation, wherein simultaneously more than one nutrients or hydrogen ions are translocated into and outside of the cell. And a good example of group translocation is PEP transferases. And the fifth mechanism is uptake of iron using siderophores. Thank you.